Hello and welcome back to BSG Webinars. I'm your host, Dan Carney. Whether this is your first webinar or you've watched them all, I'd like to thank you for taking the time to join us today. Sour beer, lactic acid bacteria, and active dry yeast with Fermentis is our 25th episode since the start of our webinar series. So if you haven't seen any of the past webinar series, I recommend you go visit YouTube and search for BSG Craft. There you'll find all of our past webinars as well as other great content from our wonderful suppliers. So please be sure to check them out and subscribe if you can. For future webinars, a link will be found on our homepage at bsgcraftbrewing.com and bsgcanada.com. We also announce them in our newsletter, which is the best way to learn about new products, important information. So please sign up if you haven't already. That can also be found on any of our homepages. You can follow us on Instagram, Facebook, LinkedIn, and we usually post daily. So we have a lot of great content coming up. So we have a couple of upcoming webinars that I'd like to announce. Next up will be Let's Be Clear on Clarity, an overview on seltzer clarification presented by BSG's Ashton Lewis. That'll be taking place 2 weeks from today at this time. Then we have Blending In with Hop Solutions presented by BSG's Eric Sanderud and Jason Hunziker of Stekdak Brewing. That'll be in 4 weeks from today. So if you have a chance, register and join us if you can. So those are already located on our website and should be ready to rock and roll. So now let's get to today's webinar. Kevin Lane, he'll have a presentation and then we'll open it up for questions. So please use the Q&A function for any questions. You can ask them during the presentation and we will doc them. And as soon as we get to the Q&A, we'll answer them in the order that we receive them. So, and if we can't get to all the questions today, we will follow up as best as we can. So now that I have all that out of the way, I'd like to introduce Kevin. Kevin, welcome.
Thanks, Dan. I'd like to thank Dan and also to thank BSG for having me back. I did a webinar for you guys. I don't know how long ago that was, a few months ago.
I think it was our second webinar. It was almost a year ago.
Really? Is it that long ago? Yeah. Sorry.
Yeah, maybe like 10 months ago.
Time flies, I guess. No, but I'm really appreciative that you have me back and happy to be back. Happy to try to educate some more brewers, more people on some of our newer products. I think today's presentation is really interesting. It's more on some of the applied research that we've done with bacteria and combining bacteria with our active dry yeast portfolio.
Well, thanks for coming back on with us, Kevin. Now that you got the presentation pulled up, we'll just keep the webinar moving. And like I said, everyone, feel free to ask some questions and we'll get to them afterwards in the Q&A section. So
perfect. Thanks again, Dan.
Over. Yeah, thank you.
I'd also like to thank everybody that's joining. It takes time out of your day and I appreciate you joining for this webinar. So without further ado, let's get through this. Today it's going to be combining lactic acid bacteria and active dry yeast for sour beer production. Again, my name is Kevin Lane. I'm the technical sales support manager for the Americas for Fermentis. I have been with Fermentis now for about 7.5 years. It will be my 8th year anniversary in November, I believe, this year. Oh, sorry. So just taking a look at our agenda today, we'll start by talking about the sour beer processes, both more traditional sour beer production, and then more focusing on what we'll talk about today, which will be the new age or kettle souring process. Then we'll get into just really briefly the lactic acid bacteria metabolism. It's really important to start there because it's one of the main points that's distinguishing these two bacteria that we have now. Then we'll take a look specifically at the lactic fermentation parameters, the pitch rate, the wort density, the temperature of that lactic fermentation, And then lastly, the aeration conditions and how each of those we can find an optimal set point or range or amount for what it is that you as a brewer is targeting in your sour beer. And it'll, I guess, slightly explain how you can adjust the parameters to achieve what you're looking for. Then the fourth section, we'll take a look at sour beer characterization, and the alcoholic fermentation performance of that kettle soured wort. Then we'll take a look at sensory analysis of those beers, and then the aromatic expression, which has somewhat of a, not revolutionary, but I guess a surprising note for Fermentis when we got into this information, into this research. And then we'll just wrap it up at the end with conclusions. So in terms of the beer production process, if you're a brewer, I hope you know this process. You start with milling the grain, then mashing at the different temperatures, then filtration or lautering, where you're moving the solids from the liquid phase. Then from there you go into boiling for a certain period of time, whether that's 60 or 90 minutes or whatever your recipe calls for. Then cooling of the wort into the fermentation cellar where you ferment. And then once fermentation is complete and you achieve a low level of diacetyl or whatever your markers are for end of fermentation, you move into some sort of maturation usually, and then go to packaging. So it doesn't have to be bottling. It could be bottling, canning, kegging to a bright tank that you serve in your brew pub, whatever the package is. Again, the bottling is just kind of packaging, I guess, in the end. So one of the more traditional souring processes is this mixed primary fermentation that can occur in or not necessarily in oak barrels. These are just examples of types of beers that are typically made with these different traditional souring processes. So in this case, we have lambic or goose that is typically containing malt and wheat for that fermentation. You go through the standard practices of milling, mashing, filtration or lautering, boiling, cooling, and then in this case, you go into a mixed primary fermentation in oak barrels. That's just kind of that typical process, and then to bottling eventually. Of course, with this, it's quite a long time from that mixed primary fermentation to bottling. Can be a number of months or number of years, depending on the beer. A second way of traditional souring beers is going to be for the types of beers that are more or less in line with Flanders red ale beers. So this is going to be a blend of pale ale and colored malts. You're going to go through, again, this traditional brewing practice from milling to mashing to filtration or lautering, going through a boiling, cooling that wort, going to a primary fermentation, and then here it's going to be a mixed secondary fermentation or maturation in oak foeders. So again, it's going to be a mix, this time post-fermentation, post-primary fermentation, and again, a bit of an aging period for this type of beer as well, and then going to bottling eventually. A third is, again, going to be containing malt and wheat. And in this is another traditional souring process where you're going through the traditional practices of milling, mashing, filtration, or lautering, and then boiling, cooling. But in this case, you're doing a co-inoculation of lactobacillus and yeast. And then from that fermentation going to maturation. With this type of process, you're usually going to be producing somewhat or something similar to a Berliner Weiss. It's going to be a low IBU, and that's important. We'll talk about that a little bit later. Lower than 5 or 6 IBU, which will avoid those compounds from interfering with the bacteria. But now today, we're going to focus on not necessarily a new age practice, but something that's a little bit more new, a little bit more controlled than those long maturation and long aging in wood or in tanks. Here we're going to be talking about the kettle souring process. So in the kettle souring process, you're going to start with milling, you're going to mash, you're going to filter or lauter, and then you'll end up with a non-hopped wort. In this case, we're going to boil that wort for a short period of time to sterilize it, to sanitize it. We'll then cool that wort, and then we'll add bacteria at this point, understanding that this is non-hopped wort. We'll add bacteria and we'll go through lactic fermentation. And in the kettle souring process, this could take 24 up to 48 hours, maybe even longer, depending on what your targets are. And then after that lactic fermentation occurs and you achieve your targets for that lactic fermentation, then you're going to return to boiling. And this process can either happen inside of the kettle in the brew house, which is why it's called kettle souring. That keeps the bacteria out of your cellar. That seems to be a more sanitary practice for the cold side of beer, but it doesn't have to. You could transfer potentially to a fermenter, perform your lactic fermentation there, transfer back to the kettle, and return to the boiling. So the second boiling, that's in the black in the traditional brewing diagram, that boiling is when you're going to be hopping. So that's a major difference for this process, is that you're lautering or filtering, bringing that non-hopped wort to a boil briefly, and then cooling that to the lactic fermentation temperature, performing the lactic fermentation, and then returning to boiling to do your hopping. And then the rest of the process will be the same. So you go from boiling to cooling, then through fermentation, maturation, into your package. So here's all four of those together. Obviously, each of them has their unique characters, their unique spots of the souring process. Some being mixed fermentation in the primary, some being co-inoculation in primary with yeast and lactobacillus, then mixed secondary, and then also the kettle sour where you're doing the souring before the alcoholic fermentation. Now, one thing that we'll talk about today, and we'll see information on later throughout this presentation, is Homo fermentative versus Hetero fermentative lactic acid bacteria. So this is something that's somewhat new to me. I wasn't exactly an expert on bacteria, nor do I consider myself an expert on bacteria today. But really defining Homo fermentative bacteria being taking one glucose molecule and producing two lactate. An example of Homo fermentative bacteria, the one that Fermentis has, is called SafSour LP 652. That is a Lactobacillus plantarum. So that's a new, I suppose, genus species. They renamed it recently, so that is a relatively newer name. It used to be just Lactobacillus plantarum. They decided to change that, but that is our Homo fermentative bacteria, again, just producing lactate. Alternatively, you have hetero fermentative bacteria. And in this case, you're not just having one glucose go to two lactate, you have one glucose going to one lactate and one ethanol without the presence of oxygen. And then if there is oxygen present, the one glucose goes to one lactate and one acetate. An example of this would be the Lactobacillus brevis. Again, that's a slight change to the name recently, or the slight change to the genus species recently. In Fermentis' portfolio, our brevis strain is SafSour LB1. So this is really the difference between the two, whether it's Homo fermentative or Hetero fermentative. Homo fermentative is just going to be producing the same acids regardless of environment, and we'll see that in the following slides. The hetero fermentative is going to be producing two different compounds from the glucose and can be somewhat controlled with the aeration rate. So now taking a look at the lactic acid fermentation parameters, again, we'll be covering the plantarum and brevis from here on out, the SafSour LP 652 and the SafSour LB1. We'll take a look at different pitching rates of the bacteria. So we'll take a look at 1 gram per hectoliter, 10 grams per hectoliter, and 100 grams per hectoliter. Then we'll take a look at how the starting gravity influences the lactic fermentation, and we'll focus on 8 Plato, 12 Plato, and 16 Plato. Then we'll take a look at the temperature and how the temperature impacts the lactic fermentation. And here we have four different temperatures that we'll look at. We'll look at 30, 37, 40, and 45 degrees. And then lastly, we'll take a look at the anaerobic versus aerobic environment and how that influences what these bacteria are doing. With each of these, we'll take a look at the acidification kinetics in terms of pH. It's a fairly good marker for the acidification of the wort, and then we'll take a look at the specific organic acids that are produced in this lactic fermentation. Sorry, so first we'll start with the pitching rate of the bacteria. So something to focus on here, I see that it goes slightly off the screen. I apologize for that, but in the upper right corner, you have the wort condition. So it's 12 Plato non-hopped wort. This lactic fermentation is being carried out at 30 degrees C, and it's all performed in anaerobic conditions. So the only thing that we're trying to control everything in a scientific experiment, trying to control everything except for that pitching rate of the bacteria and seeing how that affects this lactic fermentation. For each of these slides, you'll see that the SafSour LP652 is on the left and the SafSour LB1 is on the right. So that'll be the same for any of these pH graphs and also for the specific acid production that we'll see on the next slide. And then we'll see a lot of similar tendencies throughout these slides. That LP652 probably goes a little bit more sour, maybe a little bit faster, regardless of, or I suppose it's comparing to LB1 with the same conditions. So here we can see the 1 gram per hectoliter add rate, pitch rate is going to be in blue. The 10 gram per hectoliter pitch rate is going to be in orange, and then the 100 gram per hectoliter pitch rate is going to be in gray. And that's for both the LP652 on the left and the LB1 on the right. Here there's a dashed line, the red dashed line across the bottom is really where the LB1 is reaching its terminal point in terms of pH with the 100 gram per hectoliter. And then you can compare that to the LP652 on the right and see that the LP652 actually does drop probably down into the 3.2 something, so 3.26, 3.27. So the final pH are lower for LP652 than they are for LB1 at the equivalent pitch rates. Again, this is something that you as a brewer can control how much bacteria you're adding, therefore changing how far the pH drops. Now that pH drop is of course going to be in line, or should be somewhat in line with the amount of acid produced. We will see an exception a little bit later, but for the most part, the pH drop is going to be in line with how much acid is present in that wort. So here you're just seeing the actual measurement of organic acids in each of these lactic fermented worts. Here you can see the level of lactic acid and acetic acid after the 24-hour mark, and then again after the 48-hour mark. So the 24-hour mark is going to be in gray or silver. 48-hour mark is going to be in blue. For the LP652, you can see that it's primarily almost entirely lactic acid production. Again, that's Homo fermentative, so that's going to be producing just lactic acid from that glucose. On the right, you can see that there's a varying amount of lactic and acetic acid. And of course, the more acid production is going to be carried out by a higher bacteria add rate. So with both of these situations, you're going to see a higher amount of acid, the more bacteria is present at the beginning. That should make sense because you have more bacteria acting on the simple sugars that are in the wort. So again, LP652 produces more lactic acid than LB1, and LB1 does produce acetic acid, which you can see it's significantly more than LP652. Next, we'll take a look at the wort density. So again, it's 8, 12, and 16 Plato. Here again, we have constant variables or constant parameters of 10 gram per hectoliter, 30 degrees C, and anaerobic fermentation. Again, we're seeing similar things here between LP652 and SafSour LB1. The SafSour LP652 does reach a lower pH after the 48-hour mark. And in fact, if you trace the lines, if you were to overlay the LB1 on the LP652, the LP652 reaches quite lower a bit faster than the LB1. What is interesting to see here is that with the gravity change, we see that 8 Plato and 12 Plato are fairly similar in terms of what they reach, in terms of the kinetics of this souring, but that the 16 Plato is slightly higher. Now, that isn't necessarily a straightforward concept for anybody to think about, because you'd think that with the higher gravity to begin with, you'd have more simple sugar to be turned into the acids, which in fact is true. But what we see here is the impact of the buffering capacity of the malt or of the wort. So when you're at 8 or 12 Plato, there isn't too big of a difference in that buffering capacity. When you get up to 16 Plato, it's a much stronger buffering capacity for that pH. So in fact, when we take a look at the acid production of these fermentations, we see that the acid increases with the higher Plato, the higher starting gravity. But again, when you get to stronger gravities, you're going to have more buffering capacity coming from the wort, which is what changes that pH, which is why I said at the beginning, it's a good marker, the pH is. It's a good marker to have, and it's a good, an easier marker to read for targeting when you're kettle souring or when your beer is ready to be moved forward in the process. It doesn't really involve terribly expensive equipment where reading the specific acids and having a report on that is a little bit more complex and a little bit more expensive. So pH is a good marker, but not always perfect. And we see that here with the buffering capacity of the higher gravity wort. Next, we'll take a look at the temperature and how that influences this lactic fermentation. Again, we're looking at four different temperatures: 30 degrees, 37 degrees, 40 degrees, and 45 degrees. Again, we're trying to control all of the parameters except for that temperature. So we're doing 10 gram per hectoliter add rate, 12 Plato non-hopped wort, and again, anaerobic. And in fact, I guess I could stop here and say that we ended up making 10 gram per hectoliter our recommended add rate of the bacteria. We found that it worked quite well, and we see that throughout all of these fermentations that the 10 gram per hectoliter really did work and in a reasonable amount of time. So that ended up being our recommended add rate for the bacteria. But here we can see the influence of the temperature on that pH drop, on the rate of the lactic fermentation. And again, we see that with LP652, SafSour LP652 on the left, it does reach a lower terminal pH. On the right, we see LB1 does not reach as far. We do see similarities between the two though, and that is that both of these bacteria seem to be quite happy in the 30 to 37 degree range. The SafSour LP652 may be a little bit happier, a little bit more sour of a wort if you ferment at 30 degrees. The SafSour LB1 is more in line with each other between 30 and 37. So that's really where we come up with our recommended fermentation temperature for these bacteria, is that we find that somewhere between 30 and 37 degrees, regardless of the bacteria, you're having a really strong performance and reaching that terminal pH. Now, it's interesting to note, though, that if you go up to 40 degrees, you still have a souring. You still have a drop in pH. It isn't quite as intense. It's not quite as sour, but you do still have a souring. However, when you get up to 45 degrees, you really have little activity from this bacteria. I would make a couple of brief conclusions on this, that really changing the temperature is a way for you to control this souring. So if you want a sour beer, but you don't want it mouth puckering, not that I think any of these would be terribly mouth puckering, but you can actually increase the temperature of this lactic fermentation to adjust how fast or how far that pH drops. Additionally, we can say that these lactic acid bacteria really tend to prefer temperatures that are close to human body temperature. I guess I don't personally know what a cow body temperature is, but I imagine that it's somewhere close to that as well. But we can see that really it's preferring close to that human body temperature. And I think that that's relatively true for most lactic acid bacteria, that's going to have that preference somewhere around human body temperature. We also see that temperatures higher than 37 do impact that lactic fermentation performance, and that could be a good or a bad thing. It's one of the parameters that you as a brewer can control, if you have the capacity to, is control that temperature higher or lower to achieve the target that you want. Again, taking a look at the acids produced by either bacteria, we have the SafSour LP652 on the left, producing again primarily almost entirely lactic acid. There's a very small, little tiny bit of acetic acid. And then on the right, we have the LB1, SafSour LB1, which is producing more acetic acid. And the ratio of the two is going to be closer, the lactic to acetic. And we'll see that, I believe, in the next couple of slides. But you can see that at lower temperatures, at 30 degrees, compared to 37 or compared to 40, you have more acid produced at 30 degrees than you do at 37. And then again, more at 37 than at 40. So you can see kind of the preference of these bacteria in terms of when they're the most comfortable, when they're doing the most fermentation for this lactic fermentation. Finally, we'll take a look at the anaerobic versus aerobic environments for these two bacteria and how that influences the lactic fermentation. So again, we control all the other conditions, 10 gram per hectoliter, 12 Plato non-hopped wort. Again, remember, I guess I didn't say it completely here yet, but bacteria are going to be sensitive to the hop compounds, primarily isoalpha acids. So it's important to understand that when you're kettle souring, you want that wort to be non-hopped wort. You want that wort to be deficient. You want hops to be non-existent in this kettle souring so that the bacteria are as active and as healthy as they can be. Hops do contain some microbiological deterrents, which is good for beer, but if you're trying to sour, then you don't want that present. So it's important to understand that for kettle souring, if you're using these bacteria, then you want to make sure that there is no hop present to make sure that these bacteria do perform as well as they can and perform according to what you see here. From what I understand, the SafSour LP652 can withstand some bitterness, some IBU. If I remember correctly, it was still able to produce some acid at 5 IBU, but once you achieve 10, there really was no activity. So that could be interesting, but for all intents and purposes right now, we're talking about kettle souring. So this is without hops present. It's something that I think we'll be looking further into in the future to identify what the threshold is that the SafSour LP652 can withstand. Again, what we see here with the red lines are without oxygen, and then the blue lines are with oxygen present. You can see that really the acidification kinetics are fairly similar with both, albeit with the SafSour LB1, the presence of oxygen does delay that pH drop a little bit, possibly up to a day, or sorry, not up to a day, it's up to 4-hour difference at peak pH drop, but in the end, they're both basically achieving the same terminal pH. Now, when looking at the acid production between these two, the farther two bars in the back, the yellow and the blue, are with LP652, SafSour LP652, and the two in the foreground are with SafSour LB1. So in both cases, the bar that's farther away is without oxygen, and then the one that's closer with either strain is going to be with the presence of oxygen in an aerobic environment. And here what we see is that with LP652, really the presence of oxygen or without oxygen is not terribly affecting the acid production. There is a slight difference in the lactic acid production. When there is oxygen present, the bacteria actually produces slightly less. Again, this could be implemented as a control for this souring. But then when we look at the LB1, SafSour LB1, you can really see a strong impact. First of all, there's more acetic acid produced in general with or without oxygen. But when there's oxygen present, which is the gray line or the gray bars that are closest to us, you can see that the ratio of lactic acid to acetic acid, and that's the ratio L to A, L over A, it goes from a 3.8 to a 1.6. So really, really a much stronger production of acetic acid in relation to lactic acid. And that should make sense because when we saw the hetero fermentative, we saw that when there is oxygen present, the SafSour LB1, the brevis, actually transitions from a lactic acid and ethanol to a lactic acid and acetic acid production from that glucose. So makes sense, but that's really one of the distinguishing things here that we can see between these two is that, okay, with oxygen present with SafSour LP 652, you do have a slight reduction in lactic acid production, which could be considered a control. But then when you look more specifically at the SafSour LB1, regardless of oxygen present or not, you're producing a higher ratio of acetic acid to lactic acid, or I guess a lower ratio of lactic to acetic is how this is represented. But you can really control that acetic acid production with the presence or the lack thereof of oxygen in that lactic fermentation. So again, that's what it's saying here. The acetic acid quantity produced by LB1 is promoted by aeration conditions. So again, if you have oxygen present, the LB1, the brevis, will produce more acetic acid in relation to the lactic acid produced. So conclusions from this first part, this has all been about bacteria so far. The common features between these two bacteria are that the acidification kinetics are relatively equivalent. We saw, though, that the LP652, SafSour LP652 does produce a slightly lower pH. We saw that there's no significant impact of the wort density on lactic fermentation. There was that slight buffering capacity difference once you reach 16 Plato, but really that difference wasn't terribly huge anyways. And then we also saw that between the two bacteria, they both have a preferential temperature range somewhere between 30 and 37 degrees. However, there are significant differences between these two bacteria. The SafSour LP652, the plantarum, produces more lactic acid. Remember, it's Homo fermentative, so it's just going to be producing 2 lactic acid molecules from glucose. And the LB1, SafSour LB1, produces acetic acid, and that can be controlled. And that's the last statement, can be controlled with the presence of oxygen. So you can, I guess it can't be controlled, but you can promote that acetic acid production with the wort being aerated. Now we're going, we at Fermentis, not me personally, but we at Fermentis took this a step further, and we said, okay, we understand a little bit about the bacteria. We're always going to be learning more and more about it, more and more about how to ideally treat it for what the target is in the end. But we have this somewhat controlled environment that we understand if we change this parameter this way or that one, we end up with this acidification, this pH level, this amount of lactic or acetic acid, or a combination of the two. So we can control that. Sorry again for this overlap of text. I apologize for that. So we have the souring capacity figured out for the most part. Now what happens when we apply our yeast to that? So here we set up a new round of experiments where we did 100% pills and malt. We did the lactic fermentation. So again, we did the milling, mashing, filtration, or lautering, and then we did the kettle souring. And with the kettle souring, we did 10 gram per hectoliter for the pitch rate. It was a 12 Plato wort. We did this lactic fermentation at 30 degrees C, and we did it in anaerobic conditions, targeting a pH of 3.5. So we took that wort and we applied, when we returned to the boil, we applied hops for 20 IBU. I believe it's all at the start of the boil, so just producing bitterness. And then we carried out the alcoholic fermentation now. With our yeast strains. So that alcoholic fermentation was carried out at 23 degrees and with a pitching rate of 50 gram per hectoliter. Now with that, we are able to compare the fermentation performance, the sensory analysis, and the aromatic compounds of these resulting beers. So here we're going to take a look at kind of 3 categories, I guess, of our yeast. Maybe 2, maybe 3, it depends on how you look at it. Here for the first category, it's most of our ale yeast. So here we have POF negative and POF positive strains, the POF negative strains being K97, S33, SO4, BE256, and then 3 POF positive strains, SafAle BE134, SafAle 258, and SafAle WB06. All of these yeast strains perform similar, where we have an example, K97 on the left, where you can see that the control, the kettle sour with SafSour LP652 and the kettle sour with SafSour LB1, all of the fermentation kinetics. So you can see the darker kind of maroon colored line, the orange, and then the what appears to be kind of a peach or a faint orange. I guess depends on your computer screen. But you can see that almost all three of those lines are almost in line with each other. All of them ending with basically the same resulting ethanol at the end of fermentation. So for this category, for this group of strains, we can say that for the most part, it looks like the kettle souring doesn't affect the yeast. And if it does, barely, and the end of fermentation alcohol is going to end up the same, maybe a little bit delayed, but it should end up being pretty much the same alcohol in the end. We did, however, have one strain that showed up to be a little bit different in fermentation kinetics, that being the SafAle USO5. It is POF negative. But here you can see that the kettle souring did have an impact on that alcoholic fermentation. Could be close to a day, probably, difference between the control and the SafSour LB1. The wort that was kettle soured with SafSour LB1. But again, in the end, all three of these fermentations ended at pretty much the same alcohol. So we can say that here, the kettle souring probably does have an effect on the SafAle USO5's alcoholic fermentation, but more or less just a slowing down of that fermentation and not really affecting the end ethanol, the end result of this fermentation. And then there's a third category, which is our lager yeast. I don't know, I mean, here it's clear that there was a difference for the control and SafSour LB1 versus the SafSour LP652 kettle sour. You do see a delay, you do see a lag in that fermentation, in the alcoholic fermentation, here represented by S23. But again, in the end, they all end at the same, basically the same alcohol. So for all of our yeast strains that we tested, we don't really see a huge impact on the end of fermentation, other than the fact that it might take a little bit longer with some of the yeast strains. With most of the yeast strains, it does take a little bit longer, but really they're all ending at the same ethanol. So really it's not impacting the attenuation, just the timing of it. Here we can see the apparent degree of fermentation of the yeast strains. The control maybe has a slightly higher attenuation, but that's only by 1 or 2 degrees at the most, I think. And in some cases, like the SafAle WB06, it's the opposite, that the kettle soured beers have slightly higher attenuation. But really, all of these are pretty much in line, so within 1 or 2% apparent degree of fermentation, which means that you should end up with what would be expected from those yeast strains without the kettle souring process. Now, when it comes to the sour beer characterization, we did put this through a smaller sensory panel. I don't think it went through our full sensory panel, but we did have some interesting findings from this smaller taste panel. Here you have the aromatic and the flavor markers that we asked the people to rate. And you can see that with the SafSour LP652, there was a significant increase in acidity, and there was maybe a slight decrease in sweetness. That probably makes sense. There's going to be more acid present, so you're going to have probably a difference in that sweetness, at least in perception. But then for the most part, the rest of the shape stays pretty much the same as the SafSour LB1 kettle soured beer. This was with S33. Now, when we take a look at a POF positive strain, here at SafAle BE134, you can see the blue line again is the SafSour LB1 kettle soured beer. And you can see that there's what would be expected, some phenolic aroma and then also phenolic flavor. There's acidity present, there's fruitiness, there's a little bit of sweetness. But when you take a look at that same yeast strain, but with the SafSour LP652 kettle souring, the phenolic character is greatly reduced, if not eliminated. And you can see an increase in fruitiness and an increase in acidity. So this was something that I think was quite surprising to Fermentis when we did this taste panel, when we had these results, that something's happening here with SafSour LP 652 that's either stopping the yeast from producing phenolic compounds, or that bacteria is doing something with the precursors for phenols. So the POF positive yeast strain actually ended up presenting more or less a POF negative profile when the wort was kettle soured with LP652. So if you compare the B134 to the S33 on the left, which is POF negative, it's pretty much the same shape, with that lack of phenolic character in the taste panel. So we can say that low phenolic perception detected with POF positive yeast strains when lactic fermentation is performed with LP652, and that the SafSour LB1 really respects the phenolic character of the POF positive strains, because you see that in the right, the blue spider graph, really still having that phenolic character with the SafSour LB1, sorry. And that the acidity perception is higher with LP652 than with SafSour LB1, which increases the drinkability, and that the SafSour LB1 develops rounder profile with more balanced organic acid profiles. That makes sense because it's not just the lactic acid. So we tried to look into it further. I know that this has kind of sparked new interest for Fermentis in trying to identify what actually occurred with the SafSour LP652. And here we see the control in the SafSour LB1 kettle soured beer in terms of the 4VG, the 4-vinyl glycol. And then when we introduce the SafSour LP652 kettle soured wort, you see that amount of 4VG reduced to almost where you're at with the S33. So it truly is that with the SafSour LP652, there is a huge lack of the presence of phenolic characters. Also, there was no 4EG detected, and 4EG is really what Brettanomyces bruxellensis is going to convert the 4VG. So it's going to be from ferulic acid being decarboxylated to 4-vinyl glycol. And then Brettanomyces bruxellensis will take that 4-vinyl glycol and convert it to 4-ethyl glycol. However, with the SafSour LP652, we don't know necessarily where the ferulic acid or the 4VG has gone. Something that like I said, it's kind of sparked a new interest for us, and we're looking into that. Was something that we didn't necessarily expect coming out of the initial trials. So that's why there's that small question mark at the bottom. We're not really entirely sure where that 4VG or the ferulic acid has gone. It seems, though, that the SafSour LP652 consumes the ferulic acid during the lactic fermentation. Therefore, the POF character is not detected after the alcoholic fermentation, even with any of our strains that really produce a strong spicy character, the SafAle T58 or the SafAle B134 producing that phenolic character. So it's obviously ongoing research that we're doing. We're trying to identify what happened there, but just conclusions from this presentation. Sour beer diversity can be created by the lactic acid bacteria strain choice. So it gives you a choice, not just am I going to sour or am I not going to sour. Now you have the strain choice to make at the beginning, which then can change the outcome. And that would be the quality of the acidity produced during lactic fermentation. So if you want just lactic acid produced, then you would most likely go with the SafSour LP652. And if you want acetic acid, you'd most likely choose the SafSour LB1, given that that's the hetero fermentative. And in that case, you can somewhat control, you can't control, but you can influence how much acetic acid is produced by the aeration or the oxygenation of that lactic fermentation. SafSour LB1 produces acetic acid, and wort aeration can modulate that. Again, that's what I just said. You're able to somewhat influence that bacteria with the amount of oxygen present. And because of SafSour LP652 being Homo fermented, it's producing 2 times the amount of lactic acid when compared to SafSour LB1. The other conclusions are that the flavor impact of the lactic fermentation really is different between the two. So the LB1, due to its aromatic neutrality, we have heard from some primary experiments that it may have somewhat of a red fruit or a berry type aroma in that lactic fermentation, still yet to be proven. But we have some insight into that. But it has a more aromatic neutrality and respects the flavor and aromatic signature during the alcoholic fermentation. So with the SafSour LB1, when you choose a POF positive strain, you should expect phenolic character in that resulting beer. However, that is not true with SafSour LP652, the plantarum. With the SafSour LP652, that bacteria during the lactic fermentation does create quite a really, really pleasant citrusy, maybe tropical fruit or peach notes. It really is, in my opinion, I mean, I've only had a few beers with that, but I think it's really pleasant, citrusy, fresh character to it. I can see tropical fruit as well. And the SafSour LP652 reveals the potential of the fruity expression of the fermentation. And that's more in regard to the SafSour LP652 somehow interfering with that POF character. That because when phenolics are present, it somewhat shadows the rest of the flavor development of that yeast strain. So when you eliminate that phenolic character, you can really appreciate the fruitiness, the other fermentation flavors from whatever yeast strain it is. So I believe that that's all that I have for you today. I welcome Dan to come back and drill me with some questions.
All right. Well, thanks again, Kevin. That was that's a lot to take in. So hopefully we can recap some of this with some of our attendees and hopefully people that watch it on YouTube later on. So yeah, we encourage you send some questions in right now. We are open for them. So right now, I've got from Sean Stafford. I'm making a Berliner Weiss this weekend using acidulated malt. Will residual lactic acid remain in my mash tun, possibly affecting future brews? If yes, what's the easiest way to eliminate it in my mash tun?
I mean, if you have lactic acid in your mash tun, I don't think it's a big deal, given that you CIP in between that brew and the next brew. With even a standard CIP, that should eliminate the lactic acid that's present. So as long as you're following good brewing practices and running a CIP, even if it's just the caustic CIP through that and the rinse water after, I think you should be fine with eliminating the lactic acid. It shouldn't affect the following brew. Especially, I mean, if you're, remember that bacteria is going to be very similar to yeast, that it does perform at a warmer temperature, but it also does have a thermal tolerance. So once you're going up too far above that, 37, 40 degrees C, you saw that the bacteria stopped working. So, if your concern is the lactic bacteria coming over to the next brew, really, I wouldn't see too much concern, especially, with standard cleaning practices. Additionally, the dilution of the lactic acid that would be on the walls of a lauter tun or the walls of the kettle really won't impact the next beer, as long as you're not transferring a significant amount of liquid from that kettle souring to the next batch. And remember that these kettle souring, if there's concern of the bacteria carrying over, the kettle souring that we showed, and this is at over the course of 24 and then 48 hours. So if you're going from one brew to the next brew, you're most likely not going to be sitting in that comfort zone of the bacteria for longer than maybe 5 or 10 minutes. So it really shouldn't develop too much lactic acid or acetic acid, depending on which bacteria it is.
Excellent. Can you explain the hard stop at pH 3.2 that is in some of the advertising for these bacteria?
I think it's more or less what the bacteria can handle. It's similar to, I think in relation to yeast, it's like an alcohol tolerance. So the yeast will be producing alcohol, obviously, during fermentation. That's why we use yeast in beer. I mean, of course, it produces great flavors as well, but, the yeast is going to be producing alcohol. The bacteria is going to be producing acid. And at some point, the bacteria is going to produce too much acid for it to continue producing more, similar to yeast that will eventually produce too much alcohol for them to continue producing alcohol.
Right.
Bacteria, their metabolic process is quite a bit faster than yeast. So here they're achieving really their terminal pH in 48 or maybe a little bit longer. It could be longer, of course, depending on the environmental conditions, but the bacteria is achieving that within 48 hours, really. I mean, it could be a little bit longer. Yeast, on the other hand, could take 2 weeks for them to achieve the alcoholic fermentation that really produces an alcohol level too high for them to survive anymore. So, I think one, it's bacteria being able to withstand that much acid, and two, the bacteria metabolically moving so much faster, producing more acid in relation to yeast producing alcohol that it reaches that in that short period of time.
Yeah. So our next question, do you recommend harvesting the soured wort to reuse for another batch?
It's a great question. It's not something I can recommend or tell you not to do. That's really your preference. I can tell you that the reason that Fermentis got into this kettle souring bacteria was, number one, if you know any of us, we all like really interesting beers. So, of course, there's some personal drive for us to do that. But secondly, it's a way for us to help the brewer to control that souring process. So instead of carrying over a volume of liquid that you could probably do a cell count on, but you don't know necessarily how fast that will react the next time, here we have a product that's in dry form similar. It's actually, I believe, freeze dried, but it's in dry form for stability, and you weigh out the amount of bacteria you want and you apply it to the same environmental conditions, and you have a consistent souring process. So rather than waiting for 4 or 5 days for this kettle souring to occur, because you're not sure exactly what the parameters should be for that second batch or how much liquid you have to carry over from that previous batch, to have the same process. That's really the second reason why Fermentis introduced these bacteria, is to help you as a brewer, number one, have a consistent process where you're counting on us to supply the bacteria in the same state each time, so you have the same kettle souring. And number two, that you can really control your process down to 24 or 48 hours, where it does interrupt the brew house if you're keeping that souring, you're keeping the lactic fermentation in the brew house, but 24 or 48 hours isn't like a 4 maybe 10-day souring that you just aren't sure. So really I can't say that you shouldn't necessarily do that. I mean, it's something you could do, but you're not going to have that consistency. You're not going to have that repetitive or repeatable process that you do when you use the bacteria from our package.
We actually did have one more come through. How is the pH of a soured batch affected if you blend that batch later on? So if you took half of a soured batch, blended it.
That's a great question. I don't have like
a clean fermentation.
Right. I mean, I don't have an exact answer because there's a lot that goes into that, and you saw with the difference in gravity and how that affects just that souring. And if you were to apply half of a batch of that to half of a batch of any of those 3 starting gravities, it would result in a different outcome, right? Because there's more buffering capacity the higher the starting gravity that you blend in. So,
right.
I don't think that there's a linear relationship there, and that I could say, if you do 50-50, you end up with this. I think in that situation, if that's what you're wanting to do, I think the best advice I have for you is just to do benchtop experiments and see when in what ratio do you hit the target that you're looking for.
Right. Yeah, just bench trial it and aim for what you want then based off of that. So, well, again, appreciate you joining us. We're pretty much almost out of time. So again, I'd like to thank you, Kevin, and remind everyone that There's a lot of past webinars. Obviously, this is our 25th, so a little bit of a milestone. And those are available on YouTube. And you can subscribe if you're not able to join us when we post new ones. You'll see them come on. And also, you can find the new ones via our newsletter, social media channels, or even ask your customer sales representative, and they should be able to help you out. So, Kevin, great seeing you. Hope we actually get to see each other in September at CBT. So
yeah, and thank you for having me again. I appreciate it. And I'm always happy to come back and talk some more. Not that I like my voice that much, but I enjoy spreading the information that I can.
And if it's all right with you, are we able to share your presentation with the people that were on today?
I think that should be fine.
All right.
I'll check with Steffi quick, but I'll get back to you as soon as I can.
Okay. Well, perfect. I mean, if not, they can view it on YouTube and page through either way. So,
sure.
But yeah, look for an email after this webinar. Once we post it on YouTube, there'll be a link there if you need to rewatch it. And if it's all okay with Fermentis, we'll have the presentation on there as well. So again, thanks for joining us, everyone. And Kevin, thank you very much. And we hope to see you all soon.
Take care, everybody.
All right. See you later.
Bye.